Can you hear me? Okay. Hi, I am Molishree Joshi, and I am here to do a 5-minute advertisement for my shared resource. I represent the Functional Genomics facility, which is one of the Cancer Center's shared resource. And we provide reagents, tools, protocols for studying gene function. And there are 3 main platforms that my core supports. They are the shRNA for mRNA and protein knockdown, ORFs for mRNA and protein overexpression, and CRISPRs for gene editing. You can find more details about this on our website, or just feel free to email me or call me or stop by the lab. We've been around for a while and have helped more than 300 labs with our reagents and have supported close to 100 publications so far. So I think we know what we're doing and we're very good at it. So if you think we can help you in your research, please let us know how we can help. So the main reason I'm here is to give you a very, very brief update on some of the new services and products that we are trying to launch on the CRISPR platform. So CRISPRs are the new kid on the block for functional genomics. But I am sure everybody has heard of CRISPRs. Anyone not heard of CRISPRs? Okay, good. So we are one of the few labs in the country that has the Sanger-arrayed whole genome CRISPR library. So this arrayed library has pre-cloned guide RNA, 2 guides to every gene. And so you could use this for knocking out individual genes or using it for an arrayed screen. We also have some of the popular pooled CRISPR genome-wide libraries like the Gecko and the SAM libraries. The advantage of getting these libraries from us is that we have gone through the trouble of amplifying these carefully and then sequencing them to make sure that the coverage and representation is there. And we can provide you these libraries in formats and quantities that are usable. So you don't have to do the legwork of amplifying these libraries. In addition, we can also custom make CRISPR libraries. So if you did not want to investigate the entire genome using one of the Gecko libraries or the SAM libraries, and you have a specific set of genes that you would like to use in your screens, all you have to do is send us the gene list, and we can design the guide RNAs, synthesize it, confirm the coverage and representation by next-gen sequencing, and provide you a pooled plasmid library. And we can also package it into lentiviral suspension, if that is what you wish to get. So in the past year or so, we have done this. We've offered the genetic screen assistance service to a couple of labs. So basically, if you were interested in doing a pooled genetic screen using shRNA or CRISPRs, but you are not really sure where you would start, you can just come to us. We can help you, sit down with you, understand your experiment, and then design, help you optimize the assay for a genome-wide screen or a pathway-specific screen, and then help you throughout the process of executing a successful screen to the point that we can actually use the NGS data and put it through our algorithm pipeline and give you your top hits, which you can then validate. So if this is something that you're interested in, please feel free to reach out. Okay, last but not the least, cell engineering. We have done a lot of these projects. This slide is busy, and I'm not going to go through all of this, but it is just to to let you know that we've done a lot of these projects, whether it is a point mutation or knocking in reporter genes like GFP, mCherry, or like neomycin cassettes. We've done these in a couple of different cell lines, and we continue to update our protocols based on the latest recommendation in the literature, and we can work with you to generate a reporter cell line for you. At this point, we're still working on coming up with a financial model for how we would offer this service, but for now, we do it on a semi-collaborative basis. So if you want to create a designer cell line, I think we can help you with that. So let me know if we can help. So most of this information is available on our website, which is pretty detailed, but if you feel like you are not able to find information, as I said, feel free to just email me or come by the lab. We are always there to help new projects. And that is my 5-minute spiel. Thank you.
So while he's switching those over, thanks so much, Molly. So those are going to be quick, like, get to know your shared resource things that we're going to have in front of these Cancer Center symposia. And hopefully you'll also just visit all the websites. They're all available through the Cancer Center site. And so I'm here to introduce Carlos Catalano, who I've known for my entire 21 years here. But actually, Carlos precedes me. So you may think that Carlos recently came from University of Washington, and he did. But he actually started here 29 years ago in the School of Pharmacy, and he basically worked up the ranks, became professor, and then decided to go off to University of Washington for a little while. But like the prodigal son, he returned, and he realized the error in his ways, and he's come back. But Carlos has actually spent his basically entire career as a faculty member studying one phage, lambda, and understanding it really, really well. And so he really wants to know, for example, how is it that you could shove all that DNA into this teeny little capsid? And so he studies the motor proteins that carry those out. And so we're talking motor proteins, but these, of course, are about the smallest motors you can come by. And so taking that basic biophysical and enzymatic understanding in itself, I mean, I don't want to knock it because I think the absolute fundamental research is critically important in its own right. One does not need to search for applications. But I think as Carlos has gotten long in the tooth, he's realized that maybe this could actually have some use for clinical applications. And so he's realized that, by modifying these particles on the outside and by basically shoving inside the particles pretty much whatever you want, you can have fantastic little nanoparticle delivery systems and perhaps diagnostic systems. And so he's really ventured into a new area that I think has really important implications for cancer. And so that's what he'll be telling you about. So, Carlos, are you going to use the mic or are you going to use this mic? Yeah, the portable one allows you to wander.
All right. All right. That seems loud. I'll put it way down here. Thank you. So this talk actually started with a coffee session with James, talking about this system that we've kind of stumbled upon and what could we do with it. And we went through a couple iterations of how we might do this, and one was a workshop just to talk about how we could apply the system. Another was a formal seminar. We started on that, or settled on that. But as we go through, I'd like to take this as kind of a group meeting, right? And so if you have any questions, particularly after the first part where I talk about what we've done, where we're headed, just feel free to ask, interrupt at any time. It's great to see some old faces, like James said. I've been here for a long time. I mean, some really old faces, and to meet some of the new ones that hopefully we'll get to know you as time goes on. All right, so this is me, and yes, I've spent about an 8-year sabbatical at the University of Washington, came back in 2015, and when I came back, I brought back a new project. So historically, we've looked at bacteriophage lambda. When I started this 30 years ago, I would talk to people, talk to audiences and say, we work on this obscure virus called Lambda. And there'd be this laughing from the audience, right? Because everybody knew about Lambda, of course. And now all the graduate students are going, Lambda, I better look that up. But anyways, bacteriophage Lambda. It's a bacteriophage. It's a virus that infects a bacterium, in this case, E. coli. All right? We have three basic research interests in the lab. The first is the motor protein. This is the enzyme that takes DNA, viral DNA, and shoves it into a empty capsid, a pre-assembled capsid. One of the things that James didn't tell you, this is amongst the most powerful biological motors known. Once the enzyme is done, it's got DNA stuffed into that shell at liquid crystalline density, generating an effective internal pressure of about 20 to 30 atmospheres of internal pressure. Now, to put that into context, some of you may have heard of this beverage that's got alcohol in it. It's called champagne. And when you pop that cork, if you just let it go, it goes flying across the room, right? That's about 5 atmospheres of pressure. So this virus motor packages DNA. We want to understand all the ins and outs of how this works. That project led us into a project trying to understand the box that the DNA is put into. So that led us into a physical virology project where we tried to understand the capsid assembly mechanisms. There's a variety of maturation processes that go on, and that led us to try and understand those maturation processes, how they're connected to packaging. Once we started, and remember, James said it, but I'm really a biochemist and enzymologist, biophysicist. So we want to understand how all these things work and intimate details of how the motor works, intimate details of how it's associated with the capsid. We therefore build up in vitro systems, highly defined in vitro systems that we can characterize to great detail. In the process of that, we purified all these guys, and we decided and found that we could start modifying them and building particles that might have some sort of use, some sort of therapeutic use, some sort of diagnostic use. And to answer your question, theranostic is a particle that can serve as both therapeutic and diagnostic. Theranostic, all right? Okay, so this is the system. Bacteriophage, oh, one more. Where am I? It's hard to see back there. Okay, so this is bacteriophage lambda. It's a capsid about 60 nanometers in diameter. It's got DNA packaged into it, liquid crystalline density, 48.5 kb of DNA just shoved in there. It's got this appendage called a tail. All bacteriophage have a tail. You know about eukaryotic viruses, right? They absorb to a membrane. They got to get through a membrane. Well, bacteriophage have to get through a cell wall. That's the function of that appendage. That appendage, that tail, serves as appendage to allow the DNA to enter. The whole virus doesn't enter, only the genome. Here's a picture of phage just coating the surface of an E. coli cell waiting for their turn to enter, right? Once that DNA is injected into the cell, and I love that word because it makes intuitive sense, but what does that mean physically? It's injected into the cell. What are the molecular interactions? There are people that study those molecular interactions and transport mechanisms. We don't. We start here. The DNA enters the cell, and just like any virus, once the genome has entered the cell, that cell is infected, and it kicks off a series of steps that allow for replication of viral DNA, transcription of viral DNA, and translation of proteins that make up the capsid proteins, the tail proteins, the motor proteins, anything that the virus needs to make more viruses. Then there's an assembly step. All those pieces come together to generate an infectious virus particle. In the case of lambda, one virus comes in, one piece of DNA comes in, 200 eventually lyse the cell, leaving looking for other hosts. Okay? Once the DNA has entered the cell, this process of replication and assembly is recapitulated with high conservation, not only in many of the cotoviruses, which are the tailed bacteriophage, but also in the adenoviruses and the herpes virus. So it's generally conserved across prokaryotic and eukaryotic systems. Okay, so we basically study the packaging event. That's the first project, if it'll let me go. Apparently, we don't study anything today. All right?
Is it going on to the platform?
No, it's not progressing here, so. All right. Let me try this one. Next slide. No. Okay. So enzymology, right? So this is the enzymology process. This is the packaging pathway. Okay, and this is where we started. This is the motor protein. The motor protein binds to viral DNA, It then binds to a capsid, it packages the DNA into a capsid, and then releases it such that it can have other proteins put on it and ultimately assemble a packaging motor. Or packaging a viral particle, and we're very interested in the packaging motor itself. It's a 2-subunit protein, and over the years we've identified a variety of allosteric interactions amongst the protein subunits. There's 3 ATP binding sites, one of them hydrolyzes ATP. There's a restriction endonuclease activity, there's a DNA translocation activity, there's a packaging ATPase that drives translocation. We're very interested in understanding all those intimate details of how the enzyme recognizes with specificity viral DNA and packages viral DNA. Okay, so that was Project 1. Project 2 then derives from this, because remember, this is going into a viral capsid. So that led us into some physical virology studies trying to understand the viral capsid itself. During the packaging process, this is the box that the DNA goes into. This is the nascent particle. Note, it's an icosahedron, 12 corners, 20 sides. Okay, it's an icosahedral particle. It's small and it looks rounded. It's not very faceted. It's not very angularized. The walls are very thick. This is what the motor recognizes. It binds to DNA, then it binds to the capsid. Okay? Then it starts popping DNA into the capsid. Once about 15% of that genome enters, the capsid undergoes an expansion process. Conformational change of 420 copies in that capsid change their shape, generating an expanded shell that's thinner, more faceted, it's angularized, okay, so it can accept the entire genome's length. That guy in many viruses, including Lambda, is really quite unstable. So if the motor tries to package a bunch of DNA into it, it'll break. And we know that. We've done a lot of in vitro studies looking at the expansion process, the structural aspects, and the thermodynamic aspects of expansion. How does DNA do that? Once this guy expands, though, it's a little fragile, and to get the whole genome in there, you need a decoration protein. That protein is called GPD, and we'll be talking a lot about this guy. This guy, you see the little orange guys, they add 420 copies to the capsid surface. On a icosahedron, there's 423-fold axes. 3 of these guys bind as trimer spikes to each one of those. 140 of them give you 420 copies of this protein. The protein remains monomeric in solution. However, when it sees the expanded shell, it adds its trimers to each of those 140 vertices. Okay? So that's kind of sort of where we're at with that. That's our physical virology stuff. This is what we've done with it, examined the capsid expansion by DNA packaging, DNA packaging reactions. How does the motor do it? How does it know when to start and to stop? The thermodynamics of expansion. One of the critical things we found is we could take the procapsid, purified procapsids, because we were completely purified proteins. That first slide I showed you showed the pathway, right, from viral DNA to infectious virus particle. There's about 10 proteins involved, a tail that's got 11 proteins in it, a capsid that's got 4 proteins in it. We've purified each and every one of those proteins. We can throw them all together, by lambda DNA, which you guys probably use as molecular weight standards, but it's actually a viral genome. Throw it in there, and we can isolate infectious particles from that system. So now we can understand and probe every step of that reaction. Okay, but this kind of thing, what we found out is if we take the capsids, expose them to urea, they will expand in vitro under very defined conditions. If we purify the decoration protein and add it, we can decorate under very defined conditions. And so we studied that, the structure and thermodynamic effects of that. Okay, so one other piece of data, because so far I haven't talked about anything clinical or diagnostic. Matter of fact, if you note, the title was totally different than what it's supposed to be. But I'm getting there. There is a title coming up. Okay. Lambda phage display. Anybody know about phage display? Right? Phage display, you take a virus, you take one of the surface proteins and you make a fusion protein of something you want to select for. You run it through a variety of selection processes, and you can isolate a protein that binds with very high specificity and high affinity to a target protein. Well, this has been used for phage display. It's been used to decorate and express stuff. And in the literature, it said that there were viruses, and this is all done in vivo. You mutate, make fusions into the viral chromosome. You mutate that guy, it expresses a protein that's displayed out, and you can select for it. One of the things that they did was they made green viruses by fusing GFP to the decoration protein. So now the virus binds, the motor binds DNA, it packages DNA, expands, decoration proteins adds, it's got a GFP fusion protein on it, so now you've got green viruses in the context of an infectious process. Okay. So one of the first questions we asked, well, now that we can do all this in vitro, can we do a decoration reaction in vitro under defined conditions? And the first test case we used was this decoration protein fused to green fluorescent protein. So we cloned this, expressed this, purified this. We have capsids that we clone, express, and purify. Can we now do this under defined conditions in vitro? And of course, this is the reaction. So we take the expanded capsid. Once it's expanded, that decoration protein will add to it. By adding mixtures of wild type and green fluorescent protein, different ratios, we can tune it to, yes, it does work, and we can do it such that we get a variety of particles with a variety of different numbers of surface densities, and I'll be talking about percentage surface densities. Remember, 420 copies of this decoration protein, we can put 4 for 1%, we can put 200 for 50%, etc. You see, I'm not a mathematician, but 200 for 50%. So we can tune this decoration to as much as we want. None, 100%, 10%, 20% GFP on this shell. And we can do that because we're not in a cell, we're in vitro, right? And this is the EMs of them. The one particle on the left is a wild-type expanded capsid decorated with that decoration protein. You can see it's a lot more angularized than the procapsid. On the right, this guy's got about 50% green fluorescent protein on it. It's green, right? The particles are green. And if you look, there's all real furry stuff along the outside. That's the green fluorescent protein projecting from the surface, okay? So now we know we can do that. Well, that's great, but phage display in vivo and this system in vitro are limited by the fact that we have to use fusion proteins. So we have to have genetic constructs that we can fuse. So now we can make peptide and protein fusion constructs, decorate the shell. That's one limitation. And it's limited to genetic fusions, right? Not only that, but if you have a big protein like GFP is relatively large, and this has been shown in phage display too, once you get big proteins, the virus have trouble because you start screwing around with the decoration efficiency, and that protein's required to stabilize the shell, right? If you don't have it there, the virus is non-viable, so you're kind of stuck a little bit. So what we decided to do is just see if we could modify this a little bit. And what we did is we took that decoration protein, and that is the structure. That's the structure of the trimer. That's a crystal structure. And in that one surface loop, there is a serine, okay, S42. And what we did is we took that guy and mutated it to a cysteine. Right? So what? What does that do? When you have a cysteine, particularly when it's the only one in the protein, you can site-specifically modify it in vitro using maleimide chemistry. Right? So maleimide cysteine, very specific, very, very high efficiency. So now we can take any molecule we want. As long as it has a maleimide on it, we can site-specifically label that cysteine in that protein and get that protein to bind to the shell and display the thing. So what did we do with it? Okay, we're just playing around. Let's see what we can do. First thing we did was took polyethylene glycol. Okay, polyethylene glycol, polymer, a synthetic polymer, totally non-biologic. But it's used a lot in biopharmaceuticals for half-life effects and antigenicity effects, these kinds of things for a lot of biopharmaceuticals. You thought, well, let's play with that. So we made this guy, polyethylene glycol with a maleimide on it. We did the chemistry. We site-specifically labeled decoration protein, okay? And we could decorate the shell. 10% polyethylene glycol, 20% PEG, whatever we want to do by mixing wild type and the PEG. There it is. Okay, you can see it's labeled. Of course, it's a cartoon. That's okay. We also took mannose. This was done with Dan Ratner in Seattle. He does a lot of sugar chemistry, so we made this linked mannose molecule. We have a maleimide on it, so it reacted with our protein. Now we have a glycoprotein. Okay, and you can do this with polymers as well. So now you can take that guy and we can decorate the shell. And we can do fluorescein. So we can label the guy with fluorescein. Okay, so now we have a fluorescent GPD. And as it turns out, what we can do, oh, by the way, down in the right-hand corner, I'll acknowledge everybody at the end, but these are people who actually did the work over the years. And Jenny Chang was a graduate student in the lab. Dan Ratner was a faculty in bioengineering. And Alexis is a graduate student in the lab. She's sitting right back there and waiting to correct me anytime I make a mistake. So I'll get that stare if I screw up her data. All right. So now the important part, though, is remember I told you it's a monomer in solution. It's a monomer in solution and only trimerizes on the capsid in the presence of the expanded shell. So if we take polyethylene glycol GPD, if we take fluorescein GPD, if we take mannose GPD, if we take GFP GPD, and put them in solution at differing ratios so we can adjust the ratio just by adding it, whatever we want, then add the shells, we'll decorate the shells with a surface density that matches whatever that ratio was in solution. So now we can decorate the shells. In this particular instance, we have decorated them with mannose, GFP, we've done it with PEG. We can put 10% PEG, 5% PEG, fluorescein at 25%, whatever we want to do, just by taking that S42C, modifying it with maleimide of choice, molecule of choice, synthetic, organic, doesn't matter, as long as it's maleimide, you can get it on there, and then you can decorate the particles with anything you want. Okay, so a semi-synthetic nanoparticle. So on the left is an agarose gel showing the capsids. On the far left, you see that's the capsid band, and it shifts around depending on what you put on it. You take the particles, purify them, look at it by SDS page, and you can quantify how much of each ligand is presented on that particle. So now we have a multipartite presentation particle. Okay? So where am I headed now? And that's an EM. This is an EM. Again, note this has got GFP on it, but it hasn't got 100% GFP, so it isn't quite as furry because we dropped the amount of GFP we put on it. Okay, so about this time, this project actually started in Seattle, and about this time I came back from my sabbatical, and Dave Ross is the chairman over in Pharmaceutical Sciences, and he had me do a presentation, so I kind of went through this stuff, and of course, the question comes up as a chair, all right, so where are you going with this? What are you going to do with this? What's next? As a chair, the real question is, show me the money. Well, how can you get funded for this? How are you going to get funded for this, right? So that led to, let's see if we can use this thing for good instead of evil, okay? So that's the next question. What can we do with this? So here's where I'd like, if anybody has any questions, any comments, any statements, if anybody says, hey, you know what? I could use that for this, that, or the other. Now would be a good time to just chat about it or give me a call and we'll chat about it. But I'll show you kind of sort of where we're headed with this, the types of things we're thinking about, what we actually do have progress on, and what's just a concept. All right? So now we've got this particle, and we're going to start calling them designer nanoparticles. So you tell me what you want, you tell me how much of what you want on that surface, and we'll build it. Okay? My father was a machinist. All my uncles were machinists, right? I was supposed to inherit the family business, but I went to college and screwed that up. So now I'm getting back into engineering all over again, right? It's just really, really tiny. So what can we build? We've got the particle. We can decorate it with wild type or modified GPDs in known ratios, defined ratios, synthetic, organic, inorganic particles we want. Okay? That's the system. So what can we do with it? Well, the first thing, let's talk about malaria. Okay? This is an easy one. Not malaria itself, but the concept. Malaria, that's a female Anopheles mosquito. You get a bite, all right? And if the female mosquito if the Anopheles mosquito is infected with the malarial parasites, they're in the sexual phase in the midgut of the malarial parasite, the mosquito, injects as it's spitting. As it's biting, it's spitting, all right? Always a pleasant thought. But in that spit comes out the parasite, all right? The first thing that happens is it infects the liver. It goes through a variety of asexual replication steps in the liver, in the hepatocytes, and that's what's up there. That's known as the sporozoite phase. It then busts out, releases these merozoites. They infect the red blood cells, okay? They go through a development process in the red blood cells. Eventually, they burst the red blood cell, releasing the merozoites that then go off and infect more red blood cells and start the process all over again. That's processed depending on the species of Plasmodia, which is the parasite, will be either 3 or 4 day cycles, which is why you get quartan and tertian malaria. Right? Every few days you go through this real severe sweating and all these anaphylactic kinds of reactions because the red blood cells are bursting as the parasites exit the red blood cell. So there's the merozoites in the red blood cell. So what? It's been a very, very difficult system to crack in terms of a vaccine. And so we thought, well, let's play with this a little bit. And this came through a collaboration with a company known PhageVax, and I can talk to you sometime about that. It's an interesting collaboration. Anyway, we've got an R21 out that probably won't get funded. I'm certain won't get funded, but we'll go back in, try it all over again, okay? And we know why it didn't get funded this time. We'll fix it. This protein, merozoite surface protein, MSP1, is a protein that's expressed on the surface of the plasmodial parasite. It has been shown to be immunogenic. Not strongly immunogenic, but immunogenic, right? So hopefully, maybe we can use this protein to develop a vaccine that will develop antibodies towards the parasite. In particular, here's the protein. It's about 200 kilodalton protein. It goes through a variety, a number of proteolytic maturation steps that cuts it up into pieces. This P38 protein is one of them. Embedded in that is a 115 amino acid protein that's been shown to have some level of antigenicity towards the parasite. The last cleavage is this P42 is cleaved to 33 and 19. That proteolysis site has been shown to generate reasonably modest responses to malaria. Okay? So we thought, well, maybe, and this protein is absolutely required for invasion of the erythrocyte by this malarial parasite. So maybe what we can do is take that P115, 115 amino acid protein, probably not going to get that as a maleimide, so we'll do a genetic fusion of that with D. Okay? The second peptide is this 20 residue MSP3319 peptide that has also been shown to generate a response. So that one's small enough that we can make a maleimide with that. So using a combination of chemistry and genetics, we're going to make these two proteins, right, fuse them with GPD or chemically modified GPD with them. Now we have a decoration protein that's tagged with two malarial antigens, okay, and make a vaccine. So what we can do, we can put each of these guys on the surface alone or in combination at various surface densities defined, and if one works a little better, we can kick that up or drop it down. Once you got the constructs and you got the shells, which we have, changing them is really, really quite simple. All you do is change the concentration you put in the assembly reaction mixture, okay? So that's the idea, okay? So we're going to decorate with a bipartite antigen system and see can we do it. There's also a sporocyte protein, the one that infects the liver, that's also probably more immunogenic than these guys. But to do that assay is very different because you've got to work with worms, and that gets you into a whole system that I don't want to go into right yet. Okay, not until we show me the money, all right? But this is what we're working on, is trying to develop this thing. Right now, it's a concept, it's a theory, it's an R21. We haven't done anything on it yet. The next thing with this, though, that remember, I said that this shell is packaged with DNA. And we've got the enzyme that packages DNA. And we can drive that DNA into the shell. We've got all that set up in vitro. We can do it under defined conditions. We can do exogenous heterologous DNA. So why not put a gene that expresses one of these malarial proteins, package it up? In this case, what we're going to end up with is a combined peptide DNA vaccine, all in one particle. And that's also in the R21.
So with this, do you expect that the virus particle itself is going to stimulate like a TLR, like an innate response?
Yeah. And particularly the DNA part of it, right? So that was one of the things that came out of initial discussions. I mean, I didn't know much about this, right? About the DNA part. I knew DNA vaccines, but exactly what they do with the immune system and why they're advantageous was kind of a learning experience with this.
Frost responses, but if you could stick a string of unmethylated CP genes, wouldn't that sort of boost the data?
Well, yeah, so the idea of linking the TLR address with the protein of interest, there's a whole host of literature.
Yeah.
When you do that, you get phenomenally better T-cell responses. The B-cells get better T-cell responses.
Yeah.
And you have a unique system to mix and match all those ratios the right way. And one of the complications of the systems tends to be how much ratio of adjuvant to antigen do you want? And if you can mix and match, that actually is...
Yeah. Well, so when I am talking about this system as a vaccine, I will always tell you that the capsid itself has immune response to the capsid, right? And so we may be able to use the capsid itself. We might be able to tag LPS on it. We might be able to tag other stuff. As long as we can get it on the decoration protein, then we can decorate little of this, little of that, and see what happens.
With respect to that, to what degree is this clean of LPS?
So Alexis has actually done that. In the initial stages, it's just coming out of E. coli and purified by typical chromatographic technologies. Alexis has actually got the kit and purified out the stuff. And we haven't used it because we haven't got any animals. It was just a prototype to say we could do it.
I think in the context of sort of experiments, you do it as being very aware of the fact that LPS contamination could be.
Yes.
I think something your reviewers will say, well, how do you know it's
Yes. Yeah. And so yeah. All of that, and again, well, I want to stress that my background and my interest, for the most part, is biochemistry, biophysics, structural biology, these kinds of things. But I realize that this is something that could have application. And part of your job today is to come up with ideas for me. No, collaborations. Can we design something that you guys might be interested in? Hey, maybe if we do this, maybe if we do that. Okay, because it's really, all of these are nascent projects. The malaria project is just on paper right now. To do it would be straightforward. To have it work is another thing. Right.
So that seems a fairly obvious thing to me, to use a self-penetrating peptide like
Yep.
cap regions of HIV.
So I've got a couple other examples, and I think just getting to what could be done, let me just get through the next two examples that we're currently working on. And then, we've talked about those as well. And there's actually, right when I started, there was a guy from Switzerland or Sweden who came on and gave a talk on neuropeptides that promote transport through the blood-brain barrier, right? So as long as we can put stuff on that target and allow for transport, it's a viable project. And speaking of that, I mean, just in terms of getting into a cell, that brings us to the next project. Okay, so that was one, vaccines. Can we put antigens on the surface? Can we put multiple antigens on the surface? Could we simultaneously put DNA into a peptide DNA vaccine? This is now cell targeting. Okay, and this started, actually Alexis started this project in collaboration with Dmitry Simberg in the lab, not in the lab, in the department. He was actually working with this guy, and we were looking for stuff to do, just like now, looking for stuff to do. And he said, well, hey, we have a bunch of Herceptin. I said, what's Herceptin? And he says, oh, it's a targeted therapeutic for breast cancer, HER2-positive breast cancer. You guys will know a lot more about this than I will, okay? So we said, well, this is the system, right? There's HER2. It's overexpressed in a number of cancers, breast cancers. And, HER2 is the receptor. Herceptin binds to it and inhibits the downstream effects that cause cell proliferation. So we thought, well, maybe, why don't we just, instead of taking Herceptin, take Herceptin and decorate a particle with it and see what that does? Can we then target? The original concept was to decorate with the antibody, target those cells with specificity, and then deliver something on the inside of the shell. Okay, that was the original idea. And we're still going there. So this is the concept. Let's build that particle. Well, unlike the things we've been talking about so far, that all we need is a maleimide to tag that protein, now we're asking for two proteins to be covalently attached. So we had to change the chemistry a little bit. And what we did is there's Herceptin, right? And this little blue ball here that's unmarked is GPD. That's our decoration protein. So the decoration protein has a single cysteine on it, so we can specifically label that with maleimide. Unfortunately, the other end, right, the antibody doesn't. It's got all sorts of amino acids. And we can't use a maleimide because then we just cross-link two decoration proteins or two antibodies. We can't do that. So what we chose is this bifunctional cross-linking reagent. It's got maleimide on one side and succinimide on the other. Succinimide reacts with amines, so we treat that in limiting amounts with the antibody. And now the drawback of this is the antibody gets labeled in multiple places because it's got a lot of amines in it. It's got a lysines in it, right? But we got that guy, and then we cross-linked to D. So now we have a D with an antibody, or two or three, right? And in fact, I was kind of surprised this worked. But it did, okay? Because we're just modifying the antibody all over the place, and who knows how much activity you get left. All right, so here's the process, assembly of trastuzumide nanoparticles, right? We've got trastuzumide cross-linked to D. We've got fluorescein. We've got D that's labeled with fluorescein, and we've got wild type. So now we can adjust those concentrations and say how much fluorescein do we want in the particle, how much Herceptin do we want in the particle, and if we want later times, how much PEG do we want in the particle for pharmacokinetic properties, improved pharmacokinetic properties. So right now we're just talking about fluorescein so we can see it, and trastuzumide. And this is basically the particle, okay? This is a gel of the particle. Okay, so we've put 25% fluorescein on the particles, fluorescein GPD, right, 25% quantified, and then from 2 to 11, increasing surface density of trastuzumide. And you can see we've got GPE as a major capsid protein, so that's the shell itself. We've got GPD down below, wild type and fluorescein, and there's a fluorescence gel of the thing, so you can see we actually do have a fluorescent particle. And then increasing concentrations, increasing amounts, increasing surface densities of the heavy and light chain of Herceptin. Okay? And note above in particular, on line 11, or lane 11, above it, you see this ladder of stuff. That's multiple Herceptin adducts. Okay? But despite this heterogeneity, we get particles, and they're decorated. Okay, so this is an EM of the particles. And on the left is just the antibody by itself. So you can see, you can actually look closely and see these little Y things in there. You can get the sizes. Then these are particles decorated with 0.5% and 30% Herceptin. And again, just like with GFP, what you see is the surface of the heavily decorated particles is really furry, and that represents the antibodies projecting out away from the particle. Okay? Do they work? Right? So the whole idea of this was to target with specificity HER2 overexpressing cells. And this is, I got the recent data, so don't get mad at me, all right? Here it is. This is Herceptin all by itself, the therapeutic. Binding to HER2 plus, which means overexpressing HER2 cells, and HER2 minus, which means normally expressed HER2. And you see it's a drug, it's a well-characterized drug, and it interacts with specificity to those HER2 cells. Note that 2 micromolar is a huge concentration. It's above an order of magnitude higher than you see therapeutically. But we just push the system. You'll notice it's all around the surface of the cells. Okay? Now we take that guy and cross-link it to our particles, and what do we see? Again, we have specificity. The HER2 minus cells are not binding the thing, which would you expect. But interestingly, the HER2 plus cells are not only binding, but they're internalizing the particle. Okay? So now we've got this thing, and this I think is 4%. There is a concentration or surface density dependence on internalization. The concentration of Herceptin in this thing is what, about 2%? 30 nanomolar, excuse me. 30 nanomolar versus 2 micromolar. Okay? So now we've got a system where we can specifically target and internalize particles into a cell. Okay, with specificity. What have I got next?
No, he asked me to correct you. That's 30%.
It said 4 there. Oh, that's your fault. It says 4 there.
I corrected it on the new image.
Oh, it's always the new image. I always am missing the new image. Okay, this is 30%. Okay, and one other thing that's of interest is Alexis assures me that as you change the surface density from 0.5% to 1% to 2% to 4% up to 30%, particularly at the higher surface density values, we seem to go from just a general dispersion of the particles around the cytoplasm to start to interact with the nucleus. We see these big blobs around the nucleus. So now we may be able to not only get it in, but specify where it goes by using surface density. Okay? And it's 30%. All right? So at this point, what Alexis is going to be doing is looking at these particles. Do they affect cell proliferation in a manner similar with Herceptin? Do they affect the toxicity of cells that appear to be affected by it? How does it do to signal transduction pathways, and what does it do to metabolomics? Cell metabolism, there's a metabolomic studies that she's doing in collaboration with Angelo, and then Arthur Gutierrez-Hartman, we're collaborating with the initial Herceptin studies. Okay? Well, remember I said the original idea was to develop a particle that we could target and deliver. So what would you want to deliver? Well, because it was Arthur's collaboration, this ETS protein. Okay, ETS protein is an internal transcription factor. It's associated with mammary tumorigenesis. ESC1 knockdown appears to inhibit transformed phenotype. So the question, can we specifically target ETS inside a HER2 overexpressing cell? And the idea is, and once these initial studies are done, we'll be moving on to that, is to package up DNA into these particles, DNA that would encode for siRNA that's specific for ETS. Okay, so we'll see if it works. I don't know. The first part up to this slide, it's all been done. It all works. It's great. This part is the next question. Okay, can we now deliver with specificity a protein that, or a system that then inhibits a process? Okay, so limitations, right? Limitations. I told you the cross-linked antibodies are messy. They're messy, right? Because you get multiple adducts. They're all over the place. I don't like messy. right? I like very defined systems. I spent my whole life trying to get defined systems to do stuff so you can characterize it. And this antibody thing is messy to me. So, just all sorts of addicts. And like I said, I'm surprised, but it works great, despite the fact that it's messy. So that led us into FABs, perhaps, to simplify the system. Affibodies, monobodies, these are just small proteins that can be used in selection processes to target a protein with high affinity and high specificity. About this time, I read a paper and was put in contact with this guy, Satya Sidhu, who works up at University of Toronto, and he's developed monobodies using ubiquitin. You're all familiar with ubiquitin as tagging proteins for turnover or regulation or this sort of thing. Well, as it turns out, it's a small protein, highly plastic. You can mutate the hell out of it and it still folds. It doesn't have any cysteines in it, which is great, because now you don't have to worry about disulfides. So what he's done, he's used M13 phage instead of lambda, and he's done selection processes. So he gets the virus to display ubiquitin, it's been completely randomized, selects a protein target, and then searches for that, searches for an affibody, searches for a ubiquitin monobody that binds with high specificity. And this is just phage display for those of you who don't know what it is. You randomize a protein, you just pour it over the thing you're trying to bind, and you get higher and higher stringent conditions for elution of the proteins. The viruses that don't bind, amplify them, et cetera, et cetera. So using this system, using a highly randomized ubiquitin displayed on a virus, searching for those particular randomized ubiquitins that bind with high specificity and affinity, he's generated a whole bunch of stuff. So these are the targets. And so now he's got ubiquitin variants that bind with really high specificity and affinity for these proteins. Okay, so what do we do with it? Well, one of the ones he's got one for HER2, by the way, which I really want to start playing with that and see if we can get away from the antibody and still target HER2, or HER3 for that matter. Huh? I know. We don't have HER3, we have HER2 though. So which one do we do? Okay, so we just chose this one. I said, Dev, what do you want to work with? I mean, we're open here. What's good for you? So he sent us this one. It's ubiquitin UBV 7.2. It specifically targets USP7. USP7 is a deubiquitinase within the cell that has a variety of effects in the cell. Overexpression is associated with cancers, blah, blah, blah. So we said, okay, well, let's play with that one. From our standpoint, it's an engineering project, right, that has ultimately significant application if it works. So here's USP7 is involved in P53 metabolism. It's involved in MDM2 metabolism. So it's become a hot target. Okay, his ubiquitin variant works really well, but he can't get it into the cell. That's where we step in, right? Okay, fine. So let's see if we can get this thing tagged to D, the decoration protein, get it on the surface of a shell, and then get it into a cell. And so what we've done, again, we have to come up with a different type of chemistry because we can't cross-link with specificity. In this case, this is a small protein, no cysteine, so we engineered in a cysteine. We got D with a single cysteine. In this case, what we did is take maleimide click reagents, tagged each one of those with a donor-acceptor click pair, clicked them together. And so now we've got a click chemistry to put these two proteins together. Bottom line, it's exactly the same as we've done with everything else. Just the chemistry is a little bit different. Okay, so now we've got this D construct. And this is Winton McClary. He's in the audience here too somewhere, hiding. All right. He's postdoc in the lab, and this is his project, is to make these ubiquitin cross-links, make them possible. So he's got this guy. The plan now, so he's shown that you can do the cross-link. He's optimized that. He's shown that the cross-link GPD still interacts in ELISA, so we haven't lost functionality, the ubiquitin. He's shown that you can decorate the protein up to 5, 10, 15%. After that, and it's the same true with trastuzumab, after we have some solubility issues at really, really high surface densities, but we can get it up there. So this is the concept. Decorate with trastuzumab, probably 1%, because Alexis has shown that's really, really good for getting into the cells. If we can do that, and ubiquitin doesn't screw up the internalization, now we've just delivered ubiquitin to the cell, right? And that ubiquitin variant can then target with very high specificity a protein on the inside of the cell. So we're hoping to affect, in a rigorous and determined way, dosage-determined way, P53 metabolism within the cell. Okay, that's the hope. Okay? So summary, all right? What's my summary? Fluorescent particles. So we can track the fluorescent particles. We can see them moving around. So there's our diagnostic possibility. We can combine peptide and DNA vaccines, malarial antigens, any kind of antigens to generate any kind of vaccine. Cell targeting nanoparticle. Herceptin is an antibody. We could use other antibodies, right, in a similar fashion to target cell of choice, as long as there was specificity there. Specificity of targeting intracellular. This last bullet point was, we got a small grant at the University of Washington, and the title of it was Intracellular Delivery of Biologics. I had no idea what that meant at the time, right? I just played with phage particles. As it turns out, this provides that very possibility. Intracellular delivery and targeting of specific biomolecules and processes within the cell. So we're pretty excited about that, okay? So now you think it's done, but wait. I told you that one of the purposes of this was to be able to decorate the surface of the particle, target it to a specific cell line, and then deliver something. Okay. What I haven't told you about is our second project that I alluded to very early on, and that's what Crystal Davis is working on. She's a structural biology student, is building particles. Okay, so we can build particles, and to build particles in vitro, because everything I've told you about so far, we've got operons that express all the capsid proteins, and we purify them out of E. coli. Well, if we purify these two proteins, it's a scaffolding protein and a capsid protein. Okay? The capsid is made of 420 copies of that purified capsid protein, but if you purify that thing, put it in solution, it aggregates and forms all sorts of stuff. Scaffolding protein is required. It's a small protein. It's a chaperone protein that chaperones that protein, the capsid protein, into a icosahedron. In the absence, it goes everywhere. In the presence, it forms nice icosahedral shells. We've shown that you can make genetic fusions of these things. Is that what the next guy is? Okay, so here's our particles. And Maggie, there's Maggie right there, did a lot of this work in Seattle, okay, looking at capsid assembly and that role of that chaperoning protein. And I'm stuck again. Okay, okay. So if we take that, so when the capsid is filled, you got this icosahedral capsid, 420 copies of the capsid protein. Inside is 200 to 300 copies of that scaffold, right, that chaperone the process to begin with. But there are holes in the shell at this point, that scaffolding protein leaves. If you make fusions of that protein, they become too big to leave. So you just trap that inside the shell. So we've got the same kind of thing. We've got all sorts of vectors that Jenny Chang made to do facile cloning into that scaffold protein to make a fusion protein that gets stuck on the inside. It'll assemble a particle. Scaffold leaves, the other things get stuck in there, so now we've got things that we can put on the inside. In addition, we did the same thing. We put a sol cysteine on it, so now we can use maleimide chemistry to put anything we want on that scaffolding protein and hopefully get it trapped. Okay, so now we can put small molecules as fusion proteins, peptides, proteins. Hopefully small molecules that we tag, synthetic polymers, quantum dots. That's one I want to play with, is get a quantum dot in there. There's a lot of utility for quantum dots, highly fluorescent, all that sort of stuff that goes with them, but they're relatively toxic, right? So can we put it in a box that protects it and then target it for diagnostic purposes? Contrast dyes for various types of radiology kinds of things. These are possibilities. And on paper, they all look great. Right? We haven't actually done many of them, but this is what we're trying to pull together. Okay? So now what we have is a nanoparticle design system. That's what it is. So now we can build stuff, bake a shell, put what you want on the inside, decorate with what you want on the outside. So you tell me what you want. That's what we're calling designer nanoparticles. Various applications. They form these icosahedral shells. Since they're semi-synthetic, part organic, part synthetic, we call them the Borg particle, and that's what we're trying to sell. That's what we're trying to do. That's what we're trying to find a good utility for, right? Anything is open at this point. We just don't know what will and won't work. And the only way to find out is to just do it. So that's what I got. And of course, I have to thank people who don't sit at their desk all day making things up, but actually making these ideas realized. And this is in the lab right now. Chin Yang, who does a lot of the motor stuff. Alexis, who we've talked a lot about in this Herceptin project. Crystal Davis works on the capsid assembly and maturation and all the thermodynamics and structural aspects of that. Winton is working on the ubiquitin stuff. And then a host of people who've worked on this over the years. And that's really all I got. So did I answer your question? I got more time.
It's potential.
Yeah.
I think an efficient cell delivery is really critical.
Yep. Well, the Herceptin stuff seems to get in very nicely. Now, that brings up a whole new basic question, and the scientific question is, how is it getting in, right? Certainly, it's getting into, we know that if you take phage, just phage, infectious phage, and you clone into the genome a eukaryotic gene under eukaryotic promoter, you will get that DNA expressed into protein. How that happens, nobody has the faintest idea. How it gets in, how it gets, if it's endosomal, how it gets out of the endosome. If it does get out of how it gets out of the capsid. How does it get to the nucleus? Unknown, but it happens.
How many signals, how much of the DNA of the lambda phage is needed to package?
None. We can put in 270 base pair pieces of DNA and they'll go in.
So you have 48.5 kb that you want.
Yes. And so for virus, for the phage, there's a limit because it needs its genome, right? Lambda was used, one of the first vectors to be used for cloning, right? There's this non-essential region. And I think it's about 12 kb of DNA in the middle of the chromosome that you can delete and the virus still grows. It's called the non-essential region. Well, my guess is it's essential in the wild, but in the lab it's fine. Okay, so it's a non-essential, and then you've put in protein of choice, DNA for protein of choice. But you're limited to that 12 kb. In the case of this system, we can package any kind of heterologous DNA in because we don't have to make an infectious virus. Yeah?
Thank you for that presentation. I was thinking the bacteriophages are everywhere.
Yeah. Was it the most numerous biological entity on Earth?
I heard 10 to the 27.
Yeah. An order of magnitude more than bacteria. Anyway.
So with that, I was thinking, when you create these encapsulated viruses, can they have horizontal transfer with other bacteriophages or other?
No, because it's not a phage, it's a particle. So we're missing the tail. What we're putting in is genetic material that has nothing to do with the phage. So it's a non-replicating particle, which is, again, another advantage of it, is we could hopefully be able to target it to a cell, get it in, get the DNA in, but the DNA has nothing to do with replication of the particle.
I totally understand that. That the other phage, that is, the ones that are naturally unmodified, can they, not by replication, but connect and transfer genetic material?
In bacteria, yes, but not in eukaryotic cells. They can't even, I mean, like I said, they do get in. The efficiency of that is unclear. I don't think it's very good. That's why targeting would be good. But yeah, they're non-replicative particles in eukaryotes. In prokaryotes, they will replicate, and that's a major issue for horizontal gene transfer. It's mediated to a large extent by phage in bacteria. And it's also a big factor in antibiotic resistance. In toxin, cholera toxin is a phage-encoded disease. Diphtheria toxin is a phage-encoded disease. So there's all of that that's going on. And phage can complement and cross over if you get multiple copies in. But again, that's prokaryotic cells.
So I think one advantage of the system is probably that you can deliver so many different things.
Yeah.
You know, if you target one thing, a cancer will evolve around it.
Yeah.
Just like HIV with one drug. And it took 3 to limit that evolution for HIV.
Yeah.
And probably the number for cancer cells will be on that same sort of relative
At
ballpark.
least, yeah.
So do you think that's quite feasible that you could effectively target multiple things at once?
Well, that's what I was saying about this Herceptin ubiquitin variant, that one targets HER3, one targets HER2. Can we put them both on there? Because that's what HER2 does, is it interacts with HER3. Now you've got 2 of them targeting 2 things in the same pathway. Then it gets internalized. Then can we target an internal protein, right, using a different ubiquitin? I mean, surface decoration is trivial, right? We can get it on there. Now, whether it's soluble, whether it affects entry and all those sorts of things, obviously it's going to be system dependent. Packaging DNA is straightforward. We've done that for years, right? Now, building those particles where we have scaffolds, modified scaffolds, is going to be, we don't know for sure yet if that'll even work, because there's going to be a size limit. There's going to be, how do those proteins, when you put them in the assembly reaction, affect polymerization reactions, and we don't know. Winton has done some preliminary data and shown that if you put in glutathione S transferase, it's incorporated into the shell. We make a little bioreactor. That's the basis for making another fusion protein with rhodinase. Rhodinase is a protein that's used that detoxifies cyanide, and it's been examined for cyanide toxicity. Half-life is horrendous. It just goes away. They tried putting it in red blood cells, which works, but then you can't get the reducing thiol into the cell. So this is perfect for that. So we're going to build a fusion protein that's got rodinase in it, build a particle that sequesters the thing from, then the concept is once you build the particle and decorate the particle, it doesn't matter what you put on the inside because the cell won't see it. The immune system won't see it. Whether that's true or not, we don't know yet. But in principle, once you build a particle, it's the particle, and everything you put on the inside is immaterial.
Now, but since they're immunogenic on the outside, too.
Oh, yeah.
An application, in other words, is this going to be a one and done? I mean, could you treat once? Because the next time you treat, is the body just going to have mounted such a potent reaction against it? It's just going to...
Well, so that is a major issue. With phage and with nanoparticles, PEG has been shown to have some effect. Will that have an effect in multiple doses? It's not clear because they only do the one dose, right? Half-life this, you pegylate it, half-life this. But it's something that's used in pharmaceuticals all the time, in biologics all the time. Now, Dr. Ankerdogge here has serious problems with PEG in and of itself, but nothing's perfect. So that's why we played with the PEG, right? And we know it changes surface properties significantly because if you decorate a shell, you have a shell decorating here on an agarose gel. As you decorate it, increase mass, it gets bigger and bigger and bigger, right? Retarded migration. When you put 100% PEG on it, it migrates backwards in the gel. So it's really changing the surface properties of that thing. So we're hopeful that that'll come. Another thing is we can grow polymers on the inside of the shell. And now you've got this little fur ball encapsulated in the particle. Can we load that with drug? And have a sustained release particle that's, got this stuff on the inside. Maybe we target it to a cell type that brings it close as it's starting to let stuff out, or maybe it'll be internalized. I mean, right now it's all ideas, except for the Herceptin, the ubiquitin, and the malaria project. Yeah.
We've all been exposed to phage for a while.
Yep.
Do we have antibodies?
Yep. A lot of them are to the tail. A lot of them to the tail. So yes, that is an issue that will always be an issue, and particularly with viruses in general. But that's why, disguising the surface is going to have to be done, whether it's a one shot or multiple. Well, one shot isn't as bad. You can do that with PEG. Multiple shots, my guess is that eventually, maybe 2-3 shots down the line, it'll change. But then, maybe you'd build a family of particles. The first round has this, and you put this molecule on, right? And then the next batch, you put that molecule on. The next batch, you put that molecule on.
Maybe use it to your advantage. You could turn the cancer cells into the vaccine.
Yeah.
Hit them with something, and so the immune system's going to go crazy on it.
Yeah. So, I mean, the nice thing about the system is it's not only tunable, it's rapidly tunable. Because once you got those D constructs, you can change whatever you want on that particle surface. Within reason. Like I said, once the things get big, they kind of start sterically interfering with presentation. Yeah?
Have you looked into packaging gold nanoparticles?
We've talked about that, we just haven't gotten that far. Yeah. We've tried, so Alexis has decorated the shell with gold, right? But packaging we haven't done, encapsulating gold particles.
For radiosensitization.
Yeah.
Mm-hmm. Yeah. I mean, but this is the thing. We can come up with all sorts of gold on the inside, quantum dots on the inside, polymers that are drug sponges on the inside. They just have to be played out.
You said
Radio-label the particles? We haven't, but that should be trivial. All right, we should be able to do that. Right, as long as we can get the radio-label D, so we'd have to add a tag. We could put ESR spin labels on them if we wanted, right? So there's any number of things we could put on. We'd have a good reason to do it, a system that would be viable to show me the money, right? So we're just playing with this right now. I mean, we're playing with this relatively aggressively. Huh? No, I know. But that's what came off the Cuba Gooding. Yeah.
All right, thanks for stimulating thought.